Yes, sir, we can see your
Yes, thank you
screen.
very much. Yeah. So thank you, Alea. I am Raghavendran. I am bioinformatics mentor and research consultant with Fine Biotech. And I will take you, I take this opportunity to welcome all of you for this webinar on metagenomics analysis and study on microbiome, and also for bioinformatics for infectious diseases. So we understood that metagenomics is a way to study microorganisms in their natural environment, dissecting community structures, exploring species richness, and investigating functional roles of these microbial communities, functional roles that these communities or different microbial communities play in many areas related to human health human health and diseases. So in this program, actually, I will discuss what you will learn in the upcoming OmicsLogic metagenomics program that is going to start by 23rd of May. And this online program will actually cover main aspects of metagenomic sequencing, analysis of 16S rRNA, and other aspects of metagenomics and microbiology research using bioinformatics. So at the end of this program, participants will be conducting independent or group projects analyzing data mainly from public domain, and to learn how high impact biomedical publications are possible on the microbiome subjects. So let's go over the syllabus in detail to get an overview on the variety of different bioinformatics analysis, the logic behind the analysis, and different downstream processing methods you will be that you will be introduced to in this specific program. So we start with an introduction to next generation sequencing for metagenomic sequencing, and this could include whole genome sequencing, and this could also include 16S rRNA sequencing and amplicon data types also. So we first start with the overview of tools and resources that is necessary for this program, like you will be given an introduction to various courses, projects, data sets, how to access the associated resources, how to access the t-bioinfo server platform, and how to get access to different projects and different example projects that we are going to discuss through this program. And then we follow it up with an introduction to metagenomic sequencing and its application in studying microbial community in functional bacterial community analysis. So here we are going to understand about whole metagenome sequencing and how to understand or how to identify different different phylogenome or phyla or different variety of microbial community from mapping onto databases like Silva and NCBI, etc. So we will also deal with different data formats that is required for performing such analysis, and we will also understand the general flow of metagenomics pipeline, and we will also get a practical hands-on on variant analysis in t-bioinfo server, and how to visualize generally the mapping and the mapping of the reads and mapping of these reads to the whole genome reference genome. So and then we will go on to visualize 16S metagenomics data. So here we are going to understand the microbiome variability using amplicon sequencing, and we are going to also use 16S rRNA sequencing for the identification of microorganisms to study microbial community. We will also discuss a couple of data from human gut project, and also data from acne, or how to understand the data distribution of acne data from NCBI bio project. And what follows is an hands-on project example on human American gut and human microbiome variability. So here we will demonstrate you how the use of small subunit rRNA sequencing is performed and is used for identification of microorganisms. And we will also introduce you to the definition of gut-brain axis and microbiome and its effect on behavior of the hosts and different analysis, downstream analysis methods that can be performed after the completion of pipeline analysis on the 16 samplicon data from t-bioinfo server. And then what follows will be a detailed project study, an example project study, which I will also demonstrate, show it to you in the Learn OmicsLogic platform. So this is about metagenomic study on how the gut microbiome is affected by diet, for example, a high-fat diet and a normal diet, and how this diet has an effect and has an influence on the behavioral aspects of a mouse system in this case. And you will also learn hands-on practice on performing these downstream analysis in, let's say, phyloseq, using phyloseq library in R. You will also have different access to different visualization methods that is used to understand the taxonomic composition, and you will also given separate space to learn and practice phyloseq analysis code in R. Excuse me, it's not responding for a moment. Yes, I got it. And then we will complete the metagenomics courses by culminating into giving you information about how to plan a bioinformatics research project. So all that you have learned so far is going to come and is going to end up in making you develop your own project from publicly available data. We will show you, or we will guide you how to explore and how to extract data from such big data repositories, and how to carefully design analysis methods that is tailored for your data, and then how to perform this analysis as you had been learning throughout the program, and then how to get a good biological interpretation, and how to understand the biology and different phenotypical characteristics of the sample that you have been using for the project. And throughout the duration of the program and for duration of your project, what follows after the program, you will have access to mentors for discussion on your research progress, and you will have access to mentors and different people for troubleshooting your technical support on a regular basis, and you'll also get suggestions and expert feedback on your progress whenever it is required. So let me help you, or let me give you an example by showcasing one of the projects that we recently prepared on the role of high-fat diet on obesity. So before going into what are the different types of analysis possible for such a project, let me give you an overview of what is a gut microbiome and what follows with the analysis of microbial community within the gut microbiome. So in healthy adults, the gut microbiome exists in a state or in a mutual symbiosis state with this host. So this environment of the gut dictates both the composition and the functional productivity of the adult gut microbiota. And this gut microbiota may interact with the host through presentation of different ligands, such as molecular patterns that are associated with pathogens, and the different metabolites that are produced. And these different molecules are responsible, and these different molecules will modulate the immune system or immune homeostasis of the gut gastrointestinal tract. And as you can see in this comparison, the gut microbiome will have an enormous impact on the life and also on the stages of the host. So it plays a fundamental role in actually shaping the composition and function of the gut and the way the food is processed, and this becomes key factor in determining the amount and the type of material that is reaching the host's circulatory system as through the growth and production of microbiota metabolites. So in this perspective, we can see that the relationship between symbiosis and dysbiosis of gut microbiome in a healthy individual and an obese individual will differ by their, for example, difference in the variability or difference in the different types of microbiome that is available, or that is that one observes in the healthy human gut or healthy gut. So to study this in this specific project, what was actually performed is a mouse system or mouse model system was used to, and it was fed a high-fat diet and a controlled diet, and their response to a normal diet and their response to a high-fat diet is studied, and also some of the microbiome is actually transferred from the high-fat diet mouse to the normal diet to understand whether or not this high-fat diet microbiome that is transferred into a normal microbiome will influence their behavioral changes. To generally to learn about what sort of analysis that can be performed with this data, let me quickly stop share here and go over to the LearnOmicsLogic platform. I will find ways to where I can show you. If I can find this. Okay. Okay, can you see my screen on project 13?
Yes, sir, we can see your screen.
Yes. So after performing background statistical analysis, we can get a lesson on how to perform hands-on, how to set up a pipeline for hands-on analysis of the 16S amplicon sequencing data. So in this lesson, you will be guided in a step-by-step process through how to take the raw data and then how to perform this analysis pipeline and how to set up the pipeline and how to perform the pipeline and what are the different settings that is required to get the pipeline running, and what are the different outputs, output files that we are going to consider for downstream analysis. And in the downstream analysis section, these major output files are used to perform downstream analysis using phyloseq and using basic R tutorial. So this tutorial on how to perform downstream analysis on this amplicon data will include a detailed learn the code section where each and every part of the code is being explained to you, and then what follows is different data visualization based on which we can understand what the data actually tells. So here the data tells the distribution or proportional abundance of phyla between the high-fat diet and the chow diet is different for the two different mice. So that is what you will progressively see when performing this analysis using that is displayed, or that is represented, visualized using different visualization. So you also will have access to try yourself these analysis methods in a special try it yourself area. Let it load. Yes, where you can learn from this code and then perform these analysis yourself in the try it yourself section. So again, this measure of diversity like alpha diversity, beta diversity, which will also tell you how different these two mice are because of the difference in their proportional abundance of microbiome that characterizes their gut area. So you will get, as I mentioned, a detailed explanation of all possible analysis methods that you can perform, and you'll get a try it yourself section where you can try these analysis methods with specific question that is directed to you after learning the codes. So with that, I will actually pass the stage to Elia